Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today, the topic is related to antibiotics, and the title of my today's lecture is Protein Synthesis Inhibitors. What basically antibiotics are? So, antibiotics are special type of chemicals and compounds that are released by bacteria or different living organism, microorganism, and they kill or they in halt the progression, the reproduction of other microorganisms. Okay, so anti means against and biotics means life. So the basic definition is, again I'm telling you that it is produced by these chemicals, these compounds are produced by one microorganism to kill or to compete with the other microorganism in order to kill them and in order to halt their reproduction. Okay, so basically these are antibiotics. So there are different classes of antibiotics, and today I will discuss the protein synthesis inhibitors. Okay, these and how does these antibiotic works. So in the previous videos I have discussed the cell wall inhibitors. There were also antibiotics that disrupt or that halt the production of cell wall peptidoglycan. Okay, so I have discussed all the mechanism of it. In this video I will discuss that how antibiotics deal with the protein synthesis. Okay, how they stop the protein synthesis of a microorganism. Okay, so let's start with the video lecture. Okay, first of all, yeah, all the video lecture is just a schematic diagram. So first of all, you should know about the molecular biology of the protein synthesis. Okay, so I will discuss in a very simple way that how this is performed. So in molecular biology we have already studied that how protein synthesis proceed. So for example, if we are having a messenger RNA, so that messenger RNA should be placed on the 30S ribosomal subunit, and after that the messenger RNA should be placed over the 30S ribosomal subunit in the form of a special type of initiation complex should be formed. What is basically initiation complex? So when the transfer RNA, the first transfer RNA that is loaded with the formyl methionine, and the messenger RNA, they are loaded into the 30S ribosomal subunit. Okay, so they are placed over it, and after that the 50S ribosomal subunit combine with it, and the translation process started. Okay, so first the initiation complex should be formed. So the first class of the protein synthesis inhibitors antibiotics are the aminoglycosides. Okay, basically in simple it is streptomycin. Okay, different other kanamycin, streptomycin, these are the aminoglycosides. Okay, so what does they do? They do not allow the formation of the initiation complex. Means they bind to the 30S ribosomal subunit, and after that when it binds to it, so the messenger RNA cannot bind to that 30S ribosomal subunit, and after that the no initiation complex can be formed, and after that the protein synthesis the translation process could not be proceeded. Okay, so aminoglycosides have a very important and a very unique function. Okay, they blocks the formation of the initiation complex by binding to the 30S ribosomal subunit. Okay, the second is the linezolid. The second is the linezolid. So it is also having a very unique function. What is its function? It also binds to the basically it does not allow the formation of the complete ribosome when the initiation complex is formed. So after that the 50S ribosomal subunit will come and bind to the 30S ribosomal subunit, and will 70S ribosomal subunit will form, and after that the translation process will start. Okay, so what does this linezolid do? It does not allow the combination of these two subunits. Okay, although initiation complex has been formed, but what will happen? The 50S ribosomal subunit has to be will have to bind to the 30S. But what does this linezolid do? It does not allow the binding of 50S ribosomal subunit to the 30S. So there will be no formation of ribosome, complete ribosome, and after that there will be no translation process, and in simple there will be no protein synthesis. Okay, so this is the main function of the linezolid. The third are the tetracyclines. Okay, tetracyclines are very much important antibiotics and protein inhibitors. They are very broad spectrum antibiotics. Okay, it can act against gram positive, gram negative, even against the chlamydia, the rickettsia. Okay, these all tetracycline can act against it. So what is its function? It has a very unique and a very simple function. When the ribosome is fully formed, okay, when the ribosome is fully formed, so after that the loaded transfer RNA that is loaded with the specific amino acid will have to enter the acceptor site of the empty the acceptor site of the 50s ribosomal subunit. Okay, so what does this tetracycline do? They bind to that ribosome and that does not allow the entrance of the loaded amino acid. For example, they bind here. Okay, they bind here, and that does not allow the, for example, this will come and bind here. Okay, so what will it do when it is already acquired by the tetracycline? So this loaded transfer RNA cannot bind to these acceptor side. Okay, so there will be no formation of the peptide bonds, the peptide chain, and in simple there will be no protein synthesis. Okay, so tetracyclines is having a very unique function. It does not allow the loaded transfer RNA to enter the acceptor site of the ribosome. Okay, so keep this simple function in your mind. Okay, the fourth one are the chloramphenicol and the clindamycin. Okay, what are their functions? So chloramphenicol and clindamycin have a very unique function. So chloramphenicol and clindamycin has a very unique function. And what is its function? It basically stops the transpeptidase reaction. Transpeptidation reaction. In simple, if you have studied molecular biology of the the translation process. So for example, we are having this is the A side, this is the P side, and this is the A side. Okay, this is the P side of the ribosome, and this is the E side of the ribosome. Okay, the 50s. So what does it do? For example, if we are having an amino loaded transfer RNA in the A side, acceptor side, and we are also having loaded transfer RNA in the on the P side. So what will it do? The remaining amino acid that is present over the transfer RNA at the P side will combine with the new amino acid. Just look here, these two amino acid will combine to this yellow amino acid. Okay, so this is basically peptide bond formation, and it is known as transpeptidation. Okay, so all the enzymatic activity is performed by the 50s ribosomal subunit. When and in simple, these two amino acid will bind to the new amino acid and the chain will elongate. Okay, so this is the elongation process. So what does it do? The chloramphenicol and the clindamycin does not allow the formation, does not allow this transpeptidase reaction. Okay, they will bind to the 50s ribosomal subunit and will degrade or affect the enzymatic activity of this transpeptidase reaction. And these two amino acid, the existing amino acid will not cannot be bind to the this yellow nascent amino acid. Okay, so no elongation will be performed, and after that no pure or so nascent protein will form. Okay, so it is also a type of protein synthesis inhibitor. In the next slide I will show you the actual mechanism of it. Okay, now here come the macrolides. So macrolides has a very unique and a very simple function. For example, if this just look here, this amino acid, wait a minute, this transfer RNA, now it has given its amino acid to the new amino acid, and it is now free of the amino acid, so it is basically an empty RNA. So it has to move to the E side means the exit side, and it has to leave the ribosome. Okay, so then it is a type of chain reaction that first then it has to leave it, and after that the new transfer RNA will come, and then the process will go on. So when it is emptied, so then it will be released from the exit side. So what does this macrolide do? It binds to the 50s ribosomal subunit and does not allow the removal of the empty RNA, the discharge RNA. Okay, so it's a problem when the E site is acquired. So this the amino acid will not, the loaded transfer RNA cannot unload its amino acid to the new amino acids, and after that it cannot move to the E side because it is already acquired by the macrolides. Okay, and the macrolide have already blocked the removal of the empty RNA. So this all reaction will be halted, and no elongation and no protein synthesis will be performed. Okay, so this is a simple function of the macrolide. Hope you have understand this the function of the macrolide. As I have told you, and I will show you the clindamycin and chloramphenicol transpeptidase reaction. Okay, so these were the two amino acid. This is one number amino acid, and this is two number, and this was the yellow amino acid, that new amino acid. Okay, so what will it do? This NH2 will attack a nucleophile attack over these two amino acid, and this bond will be broken down, and this will bind to this new amino acid. Okay, so this is basically just look here now. This is the three amino acid. This is the for this was the first one, this is the second one, and that this was the new one. So this is all performed by the ribosomes and the transpeptidase. It is a basically transpeptidation reaction. So what does this clindamycin and chloramphenicol do? It does not allow this process. Okay, so if there is no elongation, so in simple, there will be no peptide bond formation and no protein synthesis, and we can protein synthesis cannot be performed. Okay, this is the summary of today's lecture, that which amino acid, which antibiotics can perform which function. So aminoglycoside, it binds to the 30s ribosomal subunit, and it found, I have told you the action of it, and it is bactericidal. Okay, now this is the tetracycline, it also binds to the 30s, and these all are the amino acid. So sorry, the antibiotics that are binding to different ribosomal subunit, these are their mode of action, and either they are bacteriostatic and or bactericidal. Okay, so you can use this slide in your assignments and in your presentation. Okay, so this was all about the today's lecture. If you have any question in your mind about the protein synthesis inhibitors, so you can ask me in the comment box. I will answer you there. Thank you for your concentration. If you have understand this video, so like this video, share it with your friends, and subscribe to my channel for more interesting videos related to microbiology and science. Thank you very much. God bless you.